Hello everyone, very good evening. Please confirm if I'm audible and my PPT is visible to you. Students, please confirm if my voice is audible to you. Good evening, sunshine. Am I audible to you? Okay, fine. And PPT is also visible, right? Yes, good evening, Karen. Okay, Roops, thank you for confirming all. So today we'll continue with Unit 5B. Okay. Today's topic is cleavage divisions. Yeah, we'll be learning a lot of things from here, right? We are doing CSIR question answer, net question answer discussion. Okay, so we'll be also looking at gas relation also, as we have done that part as well. Okay, gas relation, and even in model organisms, we are going to see the initial developmental part. Okay, the first question goes movement of epithelial sheet. Okay, the sheet. Sheet means as hold together some cells, right? Spreading as a unit to enclose deeper layers of the embryo is termed as what? Yes, good evening to all others. Surinder, Alexander. So we have learned some movements of gastrulation. Gastrulation is the movement of cells, right? So when we make a blastula, that's a mass of cells, a ball of cells which are together, but there is a hollow cavity inside. So the cells of the blastula will be migrating so as to form the three germ layers. Okay, three germ layers will be formed. Okay. Okay, Sunshine, Achal, you all have said three. So I had told you in class that you have to look for some keywords. Okay. Okay, Anupam, let's see. So we have to look for some keywords, because simply by um, you know just reading it as a sentence, you might get confused between two type of movements. So there are, first of all, there are two types, basic category. One is movement as a sheet, means as a layer, okay, as a epithelial layer sheet. And then there is movement of individual cells. Okay, so you have to see whether it is talking about movement of a layer or individual. So here it is individual, it is moving as a layer, and that layer is spreading as a unit to enclose deeper layers of embryo. That means enclose means that layer of cell is spreading over. So if you have a blastula, that layer is spreading over an inner layer where the deeper layers will be enclosed. Means if that layer spreads, it becomes the outer layer. Okay, so automatically the other cells, they will become inner deeper cells. So what is that movement of those layer of cells which is covering it? Yes, Achal, see, now that I explained, Surinder also could answer, Achal also could answer. Okay, and I drew the diagram also. So always draw the diagram to understand a statement better. Got it? And this is a simple part B question. Just a simple part B question. Yes. Answer is epiboly. So Anupam, Surinder, Achal, you're right. Answer is epiboly. Let's have a look at the different types of layers, different type of migration. So during gastrulation, cell migrations take place. As I said, this is group of cells, invagination, involution, delamination, and epiboly. And individual cells can migrate through something called as ingression and migration. Okay. So what is epiboly here? It is a movement of epithelial cell, usually of ectodermal nature. Ectoderm means we'll find this germ layer outside. Out of the three germ layers, that is the layer of cells, inside is endodermal layer, middle is mesoderm, and outside is ectoderm. So generally epiboly will be the outer layer movement that spread. So see here the keyword is spread as a unit rather than individually to enclose the deeper layers of the embryo. So when it encloses automatically what layer was there outside initially, so that automatically gets enclosed inside. So A is right. Now what is emboli? Emboli is another type of movement. It is also where internalization is happening. It entails movements of mesoderm and endodermal precursor from the blastula surface beneath the ectodermal layer. So you see these cells, this red color type of cells, they are undergoing emboli. So if you see the emboli diagram, this is what is the blastoderm, right? So like this, the movement is there, which is creating the archenteron. So the cells which are going inside and they automatically get internalized by some ectodermal layer outside. So the outside movement is epiboly, and the cells which are inside, which gets covered by the ectoderm, that those cell movement is emboli. So epi means top and contrasting word is emboli. Involution is where the cells inward movement. So you can see here inward movement, interning or inward movement of an expanding outer layer so that it spreads over the internal surface of the remaining external cells. Okay. And ingression is individual cell migration, where you can see these cells, they were initially present in a layer of cells, but now they have migrated in the blastocele. Okay, they have become individual cells. They are no more a part of the epithelial layer. And delamination means separation of one layer into two layers. From here, some cells are coming, as you can see, to become two layers, like how we see in avian cleavage, avian gastrulation. And here you can see that invagination, it's simple lifting up of a layer. It's simple buckling off of a layer. Okay. So let's move on to the next question. Inward movement of an expanding outer layer spreading over the internal surface during gastrulation. Tirth Patel, GATE biotechnology preparation. It's actually already going on. GATE Biotechnica is already conducting classes for GATE biotechnology. Okay, those preparations are already going on. If you wish to join the classes, then you have to contact our team. You can go to our website. Okay, number is there, contact number. Toll free number is also there, and even the office number is also there. You can contact there. They will guide you how to join these classes. And you should hurry up because February is exam and already they have started the classes. Okay, so if you want to be a part of it, you should hurry up. Otherwise, you will miss it. Yes, tell me, students. So it is inward movement of an expanding outer layer. Okay, so Surinder Saujanya, the word expanding, does it come with invagination? Think about it. Yes. Achal and Anupam, right. Okay, screen is not clear. So what you can do, Bhargavi, uh, just tap once on the screen and you will find a wheel like settings and an icon is there, settings icon. Okay. So if you click on that, you will get an option quality. In that quality, you just select higher picture quality. Okay. So it will automatically apply to your video. Try it, Bhargavi. So just go to the settings at the top corner, top right hand corner, you will get that wheel type structure. Okay. So there you just click on higher image. Got it? Yeah, Surender, now your answer is right. Okay. So inward movement of an expanding outer layer spreading over the internal surface. So this can mean epiboly also, but what is not epiboly? Inward movement. Okay, if it was only one layer spreading over the internal surface, then that will mean epiboly. Got it? So how it is not epiboly? Because of this two words. If it was only one outer layer expanding or spreading over the internal surface during gastrulation, this much would have been epiboly. Okay, but epiboly is only on the covering on the outside. Generally, it doesn't go inward. Okay, so inward movement means it is not epiboly. Now, it cannot be ingression because ingression means coming out of the epithelial layer to become individual cell. Okay, it cannot be delamination, which means separation of two layers from one cell layer of cell. So it cannot be delamination also. Okay, so it has to be involution. Yes, correct answer is involution. Sunshine, Surendra, that is right. Okay, I had already given you the diagrams. You can see here involution, how the cells are going inside. It is turning towards inside and it is spreading over another layer. Can you see here? It is spreading over an internal layer. So the movement is through the blastopore. Blastopore means this gap which is created when the cells from one end are getting pushed inside. Okay, and examples also you have to know. Okay, where and all we see them. Anyways, we are learning all this in our classes. The mammalian, amphibian, drosophila, sea urchin, all the gastrulation we are learning. Epipoly, you just have a look. The cells were square in nature, but after spreading, how their shape has also changed. Intercalation is where two layers of cells they come in between each other. As you can see now, they have become one single layer. From two layers, it has become one single layer. And another movement is their convergent extension. So where some layers of cells are there, they not only intercalate between each other, but also you will see a change in their dimension, where they elongate in one dimension and they will contract in another dimension or converge in another dimension. That's why it is called as convergent extension. Okay. So I would like to update everyone regarding the Watson Junoon Scholarship, which we have reopened once again. Scholarship program for graduates, postgraduates, and professionals. So if you want to clear your CSIR exam this December, then you must join our intensive CSIR NET exam success program so that you can achieve your academic and career goals. So your numbers are given. You can call us toll free also and um, on the office number as well. Okay, or else simply you can visit stores.biotechnica.org. Ensure that you do it at the right biotechnica spelling. You type it. Okay, and what all you get under that? You get 1 year of coaching for CSI NET and GATE exam. Even you can choose QAT, IIT, JAM or GATB, whatever you're preparing. We all give coaching for all these exams, okay? And 12 months of access to all the global and national workshops that are conducted by Biotechnica with all uh, global speakers that we have, eminent researchers from outside abroad countries in their institutions. Then our programming internship with live hands-on, project hands-on, certification courses you will get of your choice, 4 of them, and you will get direct access to coaching experts on WhatsApp also. And not to forget, you will get one VIP pass to our CRISPR conference, that is CRISPR Congress 2023. It's going to be a mega event in this year. Okay. So let's come to question number 3. In a given experiment, transplantation of micromeres. So where do we get micromeres? In a sea urchin embryo, right? We also get in amphibians, but this is regarding sea urchin embryo. Now, micromeres are present in the vegetal pole of a sea urchin embryo, that we know. So we have taken the micromeres, we have transplanted them onto the animal pole of a host 16 cell sea urchin embryo. Okay. So what of these movements will you observe? If you transplant the micromeres from vegetal pole to animal pole, In which out of these movements you will observe? So again, if you are drawing a diagram, it will be helpful for you, right? To picture. We had already seen this in our classes. So you have this micromeres, right? What is the major point of micromeres of sea urchin? That they are autonomously specified, right? They have some factors by which they can get specified on their own. And not only that, they can also instruct the cells which are present above them to become endodermal derivatives and go inside the blastula. So actually they can initiate gastrulation also. Okay. And they themselves, we know the micromeres, they are also called as PMC, that is primary mesenchymal cells, right? Mesenchymes, you know, epithelial is present in a layer of cells, epithelial cells in a layer, and mesenchyme cells are that come out of it, that is loose cells that I said. So as a nature of ingression, as a movement of ingression, these cells are going to come out of the epithelial layer and they will become um, loose cells like this. Okay, so primary mesenchymal cells, they they are going to make skeletogenic mesenchymal cells. Actually, these primary mesenchymal cells only later join and fuse together to form skeletogenic rods. Okay, so they are asking if such cells are placed onto the animal pole. Now we know this is the animal pole and this is the vegetal pole. So if such cells are placed on the animal pole, then what is going to happen? Yes, sunshine. Okay, let's have a look. So, anyways, I already gave you half the answer, right? Now, here I will tell you there is a doubt regarding the answer key for this question. Okay, now actually, CSIR is not wrong because in Gilbert book also it is written like that only. Okay, in Gilbert book, one edition also it is written that if you transplant the micromeres at the animal pole are going to invaginate into the blastocele. So, according to Gilbert, this is the answer, because of which CSIR also has given this answer. Okay, but actually, this is not the fact. Okay, no, PMC doesn't invaginate. PMC is, see, why do we call mesenchymal cell? What is the full form of PMC, first of all? It is primary mesenchymal cell, isn't it? So mesenchymal means what, Surinder? It means loose cells. We have studied in class, right? It is the unconnected loose individual cells, right? So PMC means that they will become individual cell. And we just saw the movements, right? So invagination you saw, it is movement of a sheet. So how can PMC invaginate? Got it? So that is the reasoning that I am going through. The problem is in Gilbert book is also written invaginate. But if you check for some research paper, in fact in Gilbert only, in some other place, that is in the text part. Okay, in the diagram part it is written transplantation diagram, but rest everywhere it is written that the transplanted micromeres they will ingress. Means at this side these transplanted micromeres will ingress. So if they are transplanted on the other side, there also it will ingress only. Got it? There also it will ingress only. Right? Now what will invaginate? Actually, this micromeres will tell the cells which are present above them to invaginate inside the blastocele. So if you have a look at the micromeres, right? This is our first step that happens, the vegetal plate thickening. Okay. Now see in the diagram also it is written. Even in Gilbert book also you will find like this. Okay. That initially the primary mesenchymal cells are formed. See it is labeled primary mesenchymal cells which are separated. Okay. So if they are separated means the only type of movement that is possible is ingression. So primary mesenchymal cells change adhesive property and the large micromeres start to migrate into the blastocyst as free mesenchymal cells. So that is ingression. Okay, mesenchymal cells are loose cells that differentiate into many different organs. Okay, now the formation of archenteron, there we have the invagination movement. So the first movement there is the vegetal plate invaginates. So what was here like this, now this will invaginate like this, okay, into the blastocyst. So if you are putting transplanted micromeres to the other side, from there also it will tell the ectodermal cells to invaginate inside. Okay, so from here also invagination will start. But it is not the micromeres which are invaginating. No, micromeres will come out as loose cells only. The transplanted micromere themselves will come out as loose cells only. Okay? Yes, exactly, Saujanya. Very good. It will change the fate of nearby cells. So, the transplanted micromeres are telling the cells which are present near to it, that is the previously what was ectodermal cells, now they will change their fate and now they start to invaginate inside. So a second axis will be formed because from here also axis will be initiated. Okay, so according to me this should not be correct. Okay, and actually it should be ingress, but problem is in Gilbert book also, in that diagram they have mentioned invaginate. That is why the CSIR also has given that same thing. So transplanted micromeres to ingress into blastocyst to create a new set of skeletogenic mesenchymal cells. Now third, what is said, so that's what I already told you, no sunshine, that CSIR has mentioned option 1 as answer key. They have not changed it. Okay, because in Gilbert book also they have for this particular sentence that if you transplant the micromeres from vegetal pole to animal pole, then this will happen. That's what Gilbert says. But in rest places of Gilbert, it's clearly written that primary mesenchymal cells ingress to form skeletogenic mesenchyme cells. So if you take them on the other side also, then also their transplanted micromeres will ingress only. You will find it in other research papers also. So accordingly, that second should be right answer. But since CSR has given this, I can't say number 2 here. Okay, so that's why. But the concept wise, number 2 should be the answer. Now the transplanted micromeres will mingle with host micromeres. No, not at all. Okay, because transplanted micromeres are on the animal pole and host micromeres are at the vegetal pole. So how can they mingle with each other? Okay, so this is wrong. Okay, to ingress into the blastocyst. In class, we discussed this one only, no, Saujanya? The same PPT I discussed. I mean, this same slide. You remember? So why will I say invagination in class? It will ingress. Primary mesenchyme ingress. Invagination is there, but it is later. The archenteron invaginates, that is the vegetal plate invaginates. Okay, this is the first movement. I don't have the other slides, but there also it is clearly mentioned. I showed you some scanning electron micrographs also of the PMCs coming out of this layer. Do you recollect now? And then I showed you that this archenteron lifts up by invagination, then by convergent extension, it will become thinner. Do you recollect now? Okay. So invagination comes later, after that convergent extension. Then involution will also be there. But initially it is ingression. Just check the video once. I don't think I have told anything like that. Okay. So the transplanted micromeres will form the secondary archenteron. Now transplanted micromeres, secondary archenteron, the micromeres don't form archenteron. Micromeres, even here also, they will form skeletogenic cells only. Yeah, the nearby cells, it will tell them to become archenteron. So what was prospective ectoderm before, now they will tell them to become the archenteron. Okay, fine. I hope this is clarified. Now next question. Yeah, so see here. Ability of the micromeres to induce a secondary axis. Okay, so this is exactly from Gilbert book. I told you, in Gilbert it is written. So that's why That's why CSR also have taken this. The transplanted micromeals invaginate into the blastocyst to create a new set of primary mesenchyme cells. But this is not matching with the diagram. This is the diagram of Gilbert book, and this is the corresponding explanation of this diagram. Both have been taken from Gilbert book only. Okay, but the problem is this second point is not matching with the own diagram, because you see it is written transplanted micromeals which are given in red color, right? So invaginate into blastocele. But how is this invaginating? We are clearly seeing that these are the primary mesenchyme, which is written from donor cells. Okay, it is written from donor cells. You can see here. Okay, fine, Saujanya. Okay, and it is written to create a new set of primary mesenchyme cell. How can something invaginate to create mesenchyme cell? It is not going with its own diagram, and they induce animal cell next to them to become vegetal plate endoderm cells. This will be invaginate. Okay, and it is written over here: transplanted cells induce endoderm. They themselves don't become endoderm. Okay, gene expression and structures from animal pole cells. All right. Now let's go to question 4. This is regarding C. elegans. So it's written C. elegans embryo uses both autonomous and conditional modes of specification. Conditional specification at the 4-cell stage. So C. elegans at 4-cell stage, it can be seen conditional. So I hope you remember we have a C elegans embryo, we have the P0 cell, which then further divides in asymmetrically to produce a bigger AB cell, and then we have a P1 cell, right? So then the AB cell further divides equatorially and it produces two cells, two AB daughter cells, right? And then you have the P cell which forms P2 cell and EMS cell, right? And the cell and the one closer to P2 it becomes ABP, one towards the anterior side becomes ABA. I hope you remember this is what they're talking about. It can be seen in the development of endoderm cell lineage and also in the establishment of dorsal ventral axis. So there are following statements regarding this. Tell me which of these above options are true. No one wants to answer. What is this? So this was discussed, I think, last week. So the first statement is saying if the P2 cell is removed at the early 4-cell stage. So one thing what you need as an information to solve this question is that P2 is autonomously specified. Okay, P2 gets its specification by itself. And then it will signal ABP also, it will signal EMS also. So EMS will further divide into two more cells. The cell which is closer to P2 becomes the endoderm cell. And the cell which is AB daughter cell which is closer to P2, it becomes the AB posterior cell. Okay. So, okay, Surinder, let's see. So we know that the EMS cell, it divides into two daughter cells, right? So the one P2 which is closer to EMS cell, that forms the endoderm. Now, why it forms the endoderm? So P2 releases a signal called as MOM2, and endoderm receptor is there. Okay, so that receptor, it is MOM5. It is WNT type of signal. This is WNT signal and this is WNT receptor called as frizzled. Okay, so when P2 gives signal to this cell, in this cell there is one molecule called as POP1. So POP1 is down regulated. If it is down regulated, this daughter cell automatically gets endoderm specified. Okay. Now in MS, P2 can't send signal to MS. So here POP1 is present. So if POP1 is present, it goes for mesoderm specification. Okay. So what it is saying, if P2 is removed at early 4-cell stage. If P2 is removed at early 4-cell stage, then this signal cannot happen. If this signal cannot happen, then POP1 will be present in endoderm. It cannot be downregulated. Okay. So in that case, both of them, it will be forming MS. Because only if POP1 was downregulated, then only it will form endoderm. Otherwise, it will form MS. The EMS cell will divide into two MS cells and no endoderm will be made. So this is right. A is right. Okay. So anything can be correct. So A is right means either any of this can be correct. So we have to see the other ones also. Okay. What is the next one saying? In POP1 deficient embryos, both EMS daughter cells will become E cell. Now, if POP1 is not there at all, okay. So I told you that you have this MS and E cell. In whichever cell POP1 is there, that will be MS. Whichever cell POP1 is not there, that will be endoderm. Okay. But the problem is now POP1 deficient embryo, which means that MS also doesn't have POP1. Okay. Neither endoderm nor MS. In fact, nothing is there. Nowhere POP1 is there. So signal or no signal, if POP1 is not there, then MS cannot form. This also will form endoderm. So in POP1 deficient embryo, both EMS daughter cell will become endoderm. So this is also right. So AB correct. Still you have to see which is correct. So either C is correct or D is correct. Okay. Now they are talking about the AB signaling. This was about the EMS signaling. Now we know that yes, you have the P2 cell and whichever cell is closer to this P2 cell, it becomes ABP, right? So why this becomes, first of all, what is the signal name? The signal name is APX, anterior pharynx excess. And what is the receptor name? The receptor name is GLP1. Okay. Development is going on in morning batch. Yeah, that's right, Anupam. In evening batch, unit 1 is going on. So next week, unit 1 will be discussed. Okay. Next week in YouTube live session, what I have, that time I'll discuss unit 1. Fine. So here and after unit 1, unit 5 will be taught to you, Anupam, in evening batch. Okay. So we have this APX and then the signal name is GLP1. So it's saying that when the position of ABA and ABP was reversed. Okay. So if their position is reversed, so whichever cell is closer to the P2 cell, okay, whichever cell is closer to P2 cell, that will receive the signal APX, because of which ABP does not form pharynx. Okay. As such, this AB daughter cell both have capacity to form hypodermis and neurons. Okay, both the cell, daughter cell, they will form hypodermis and neurons. But only ABP, only ABA has capacity to form pharynx. Okay, ABP does not have capacity. Who tells not to form pharynx? This P2 with its signal APX. Okay, so it is saying that in in embryo whose mothers have mutant GLP1. So mutant GLP1 means they won't be able to receive the signal. The receptor is mutant means signal cannot be there. So in that case, ABP is transformed into ABA cell. That's correct. Because if signal is not there, then that signal was to not form pharynx. Okay. So if signal is not there, then pharynx will form even in ABP. and it will become the same as ABA, because ABA is also forming pharynx. So even D is correct. So ABD is correct. The correct answer is option 1. So yes, Surinder and Sunshine, your answer is right. Now what is it saying? When the position of ABA and ABP was reversed. Yes. Early when it is giving signal, so when P2 is giving signal, so two daughter cells are formed, right? Two daughter AB cells are formed. So even before the signaling, if you reverse their position, so what will happen? It doesn't matter which AB daughter cell is there. P2 will give signal to that same daughter cell, okay? Means if this was supposed to be here, if you change it here, then doesn't matter. Another AB cell will come. And that AB cell also has receptor GLP-1. So it will receive the signal, and in that case, this initial cell which was here, if that comes here, that will form APP. Okay, so their fates get reversed. If you reverse their position, their fate also will reverse. What was supposed to form ABA will now form ABP, taking the signal, and what was supposed to be ABP will now form ABA, not getting the signal. Okay, now since their fate also got reversed, you change the position also, and fate also reversed. So that's why a normal embryo will be forming. Yeah, if you would have changed the position, so let's say ABP you got here, okay, and ABA you got here, and fate did not get reversed. That means in anterior you had posterior ABP, in posterior you had anterior ABP. Then an abnormal embryo would have formed. But because the fate also got reversed, so position change did not impact, because anyways their fate got reversed and came back to normal. So only this no word is not correct. Okay. So position gets reversed, fate gets reversed, and normal embryo forms. Only this word no is incorrect in this entire question. Okay, because of which this C became wrong. Everybody understood this concept and this question? So see, what we learn in class and how we are applying in question makes a lot of difference, right? So simply studying from class, simply learning from class is not going to help you. You'll have to revise also. Why revision is required? Because you need to remember the concepts while solving the question. If you forget the concept, means how will you solve? How will you apply your knowledge, right? And not only that, you have to attend these sessions also, so that your question solving skill also gets enhanced. Okay. So if you have understood, then you must answer the next question. Before that, let me show you this. So P2 produces signal which interacts with EMS cell. So that in P2 instructs EMS cell next to it to become E cell. Okay, that is endoderm cell. This message is given through WNT signaling cascade. P2 produces homolog of WNT protein MOM2 peptide. It is received by EMS cell through MOM5 receptor, which is a version of WNT protein frizzled. Result of the signaling cascade is to down-regulate expression of POP1 in EMS daughter destined to become E cell. And in POP1 deficient embryo, both EMS daughter cell become the E cells. And this is regarding the signaling to AB. P2 is critical in giving signal that distinguish ABP from sister ABA. So ABA gives rise to neuron, hypodermis, and pharynx, but ABP makes only neuron and hypodermis. So if experimentally their positions are reversed, their fates are similarly reversed, and a normal embryo will form. Okay. So this is the signal which I told you, GLP protein on ABP, and APX, it is sent by P2 blastomere. In embryo whose mothers have mutant GLP, ABP will be transformed into ABA cell. Okay. So these are your signals. MOM2 is WNT homolog, MOM5 is frizzled homolog. APX is a delta type of a signal, and notch is the receptor for that. GLP1 is notch type of signal. Okay. So now is the next question. This is from the recent most exam. Let me see how many of you can solve on your own. So the following figure represents the interaction of a 4-cell stage of C. elegans embryo. So look at the diagram and then say which of the following statements are correct. You have to pick up the right combination of statements. So you already discussed the concept. You just have to apply it. I'll give you 1 more minute. Okay, so let's see what is the right answer. The fate of EMS blastomere is autonomously specified. Is this statement true? So we have to look at the diagram. So sometimes what they will do, listen to this carefully. Sometimes what they will do is they will simply what they will do is they will pick up some diagram which is correct, means from the book or some research paper they will pick up the diagram, and based on that they will ask the question. That means it is what actually happens. Okay, but sometimes they will make up some false result. Okay, that might not be happening in that organism, but that might be a false result, so that they will give. Based on that you have to answer. Now always check the diagram. Okay, whether it is matching with the fact that you know what happens in organism. It might not be, it is not necessary that the diagram image is matching, result is matching with what actually happens. Okay, so always check this first. Now see here, it is written MS fate is this orange color and yellow color is endoderm fate. So they are asking if remove P2, if P2 is removed, then what will happen? Now we know that P2 gives signaling to the endoderm cell, isn't it? So in case P2 is not present, then anyways EMS will make two daughter cell. So whichever daughter cell was close to P2, that would have formed endoderm. But if you remove P2, then both of the daughter cell you are getting mesoderm only. See orange color mesoderm, they have said this is normal, right? And in case you are moving P2, see you are moving P2 here. Now EMS was supposed to give two daughter cell P2 here. So this daughter cell which was closer to P2, that was forming endoderm. But what it has done now is the daughter cell which is closer to P2, now this will be closer to P2. So this will form endoderm now. You can see this will form endoderm now. And the other one which is not having P2 here, because you have moved it. Okay, so that means here nothing will be there. So this daughter cell of EMS will not get anything, no signal. So that will form MS. Got it? So it is matching with what we know. What we just studied, that concept is matching. Okay, so fate of EMS blastomere is autonomously specified. So this is definitely not correct, because if that was the case, P2 position wouldn't have changed it. Autonomous means you put it in some other place also, they will form what they were supposed to form. So if you change the place, then they will still remain the same. But since they are changing here, so it is not autonomously specified. A is wrong. If A is wrong, this gets cancelled. Okay. So you have to see B and C, B and C. That means it's correct. You just have to check whether D is correct or not. Okay. So let's check B and C. The default fate of EMS blastomere is MS. That's right. Because what was signal, P2 was not giving signal to this cell, right? To this daughter cell, it is not giving signal. So this came from EMS only. So this daughter cell by default, it will form MS only. If it gets signal from P2, then it will form endoderm. So default state is MS. B is right. Conditional specification is observed in development of endoderm cell. That's also right, because the endoderm specification, it depends on P2. Only if P2 gives signal, then only endoderm is formed. Okay. So what is D? So B and C is correct, as we saw. Assuming that a receptor needs to be activated for endoderm fate, right? So we know that that receptor MOM5, it should be activated. That we know, but here it is saying assuming a C. elegans embryo where the receptor is constitutively active. Now, constitutively means what? Whether signal is there or not, it will always be active. Okay, signal or no signal, it will always be active. So it is constitutively active. It is likely to develop cells of endoderm fate only in all three of the above cases. Okay, so in all three of above cases, it is likely to develop cells of endoderm. So that's right, because if the receptor is active, you don't need signal. By automatically that is going to form endoderm, because that receptor is activated even without signal. So yes, Saujanya, very good. Your answer is right. Okay. Bhargavi, I don't know which one you have answered for. I think that was a previous question. Okay. So B and C for sure. Okay, Sunshine, that you have answered correctly. I hope it's clear now, Achal. See, how can you say option 3? Because option 3 has both A and C. So how can A and C both be correct? One is saying autonomously specified, another is saying conditionally specified. Both cannot be correct, no? Right? Either it will be autonomous or it will be conditional. Right? Okay, fine. So this was the rotational cleavage division. I said no. P2 gives signal to EMS, and then according to that it will form E cell, whichever is closer to P2. Got it? So this is what we know. The division of AB blastomer is conditional. Its division and differentiation is dependent on P1. Okay, so division of P1 is autonomous. Only the P1 is autonomous. Rest all. So why is P1 autonomous? Because of the factor PAL1. It controls differentiation of P1 progeny, and pi1 is necessary for P2, and SKN1 is necessary to control the fate of EMS blastomere. Okay. Just reminding you Watson Junoon scholarship once again. So there are a lot that you also get along with this. In case you have missed out the previous ones, it has been reopened on popular demand. So join our intensive CSIR NET exam success program so that you can achieve your academic and career goals and clear it in one go in this year only, December 2023. So to enroll for the classes, you are getting a lot of things along with these classes right now. You can call toll-free number on this or our office number, call during office hours, or simply you can go and visit stores.biotechnica.org. Okay, this is also another question regarding the same topic. So as you can see, there are a lot of questions which are coming from these topics, right? So it will be 2 MS cells. Now this one I will not tell, you have to do it on your own. So this is C and this is D. Okay. C. elegans embryo uses both autonomous and conditional modes of specification. So the following statements are about specification of cell lineage. Signals from P1 blastomere instructs EMS cell for its further development. Yes, Anupam, you can join both morning and evening classes if you are free. Definitely, why not? And for our biotechnical students, we don't charge separately for morning evening batch. You can join any batch of one particular course. So if you have selected CSR, then you can join classes for CSR only anytime, our morning batch or evening batch. Anytime it's fine. Both also you can join. Okay. You just have to contact our team just mail to them. Okay, and they will, or you can simply call also. Just tell that I'm also interested in joining morning batch. So they will send you morning batch class link also. Okay, Surinder Sunshine, let's see. So in the absence of instructive signal that you get from P2, the EMS cell will divide into two MS cell. In the absence of POP1 signal, so you know that the POP1 has to be down regulated. The prospective MS cell can take up the EFET if POP1 is not there. And the MOM2 protein from blastomere neighboring the EMS cell instructs the dividing EMS blastomere to take up MS fate. Okay, Anupam says 2. Surender also has changed answer to 2. Sunshine also 2. Yes. Very good, everyone. It is option 2. So we know here, let's correct it. Signals of P1 blastomere. This is not correct. So it should be P2 blastomere. So many times we studied now, right? So that's why A is not correct. Yes, it instructs the EMS cell, but it doesn't instruct the EMS cell. It only instructs the endoderm, means one daughter cell of EMS to form endoderm. Okay, it doesn't instruct the MS. That's why it's incorrect. In the absence of instructive signal, yes, if instructive signal from P2 is not there, the EMS cell will divide into two MS because it was P2 which was converting one of the daughter cell into endoderm. If P2 is not there, then both of them forms MS cell. So B is right. In absence of POP1 signal, so POP1 is not there means whatever MS is there, so automatically they will become endoderm. Whether signal is there or not, if POP1 is not there, then the MS also will make E, and C is correct. MOM2 protein from blastomere neighboring the EMS cell. So MOM2 is released from P2 signal. Okay. It instructs the dividing EMS blastomere to take up MS fate. No, it instructs the EMS blastomere to take up endoderm fate. Okay. So MOM2 protein is released from the P2. So that's what the signal is. Got it, everyone? I hope this topic is clear for you now. Let's move on to the next question. Following statements are made regarding animal development. Which of the following option represents combination of all correct statements? So here you can see you have got from subunit 5A also. Okay. You have got, so this is also subunit 5A. Okay. Actually, this is, you can see 5B, right? So this is regarding the early development in sea urchin. This is from subunit 5B, that is cleavage division. Okay. And this is also from subunit 5B, that is gastrulation. Because 5B has all of this. So as you can see, they have combined different concepts. So if you do selective study, that may not help in your exam. So it's always wise to study. If you are choosing one unit at Biotechnica, and in induction, we already tell you how many units you should prepare in order to clear CSIR, right? So if you're preparing one unit, you should prepare it completely. Do not do selective study. That is very dangerous for such kind of questions. Okay, let me see how many of you can answer correctly. So we have already discussed 5A before in YouTube live session. So many questions we saw. Even this B also we have seen. Cleavage division, types of cleavage division. Gastrulation today itself we saw, right? And sea urchin embryo also we have discussed the early development. So you have an idea what is happening there regarding the micromeres. Okay, let's see, Achal. Anyone else? Surender, okay. Both of you are saying 3. Let's see if anyone else can answer. Yeah, sunshine also. So yes, all of you are right. Very good, everyone. Anupam also right. Correct answer is in fact 3. So I'm very happy that you're able to solve the questions now. So is it that the classes are helping you? Because of that, you are able to give the correct answer? Yes? Kindly confirm, Achal, Surinder, Sunshine, Anupam. Are the classes helping you to helping you to give the correct answer for these questions? So see, first time you all have given correct answer for this particular question. Okay. So yes, the cell is first specified as we know towards a given fate. Okay, that's great to know, Achal. Yes, thank you, Surinder. It's really, I'm feeling very happy that you people are answering correctly because of all this. Okay, so how to know a cell is specified? I have told you that you take the cells, right? And then in neutral environment also, if you grow, then also cells should be capable of differentiating by itself. Then only we can say it is specified. Okay, that's great, Anupam. Sunshine, really good. So keep on revising, otherwise you will forget. Okay. If the cells are not capable of differentiating, then we will call them naive. That means they have not received specification yet. But if the cells are capable of differentiating by themselves, then we will call them they are specified, where they are able to grow even in neutral environment. Okay, that means they have already got the signal. That's why the first option is correct. Okay, first option is correct means you have to check whether B is correct or D is correct. Holoblastic rotational cleavage is observed in tunicates. So there we need to see our chart. So holoblastic under isolecithal, you have tunicates, which is bilateral cleavage. Okay, tunicates is type of organism, so they go for bilateral cleavage. Okay, holoblastic rotational we see in mammals and nematodes. That is our C. elegans. Okay, only then we see holoblastic rotational cleavage. So that's why B is not correct. So if B is not correct, that means A and B is answer. To confirm the other, infolding of sheet of cells is called ingression. No, infolding means it can be either invagination, or if it is a expanding infolding, then it will be involution. Okay. Then conditional specification can be observed in sea urchin embryo. Yes, just now we had discussed about the micromeres. So the large micromeres are giving signal to the cells above them to form the endoderm and the mesoderm. So they go inside and they invaginate, isn't it? They start invaginating. So that's nothing but conditional specification where they are giving signal to another set of cells. So D is correct. Okay. So we have one last question, very easy question. So we had discussed cleavage division. In the avian embryo, the blastocyst-like fluid-filled cavity is formed between what? So avian means chick embryo. So we know that in avian embryo, you might recollect that we have telolecithal egg. Okay, so telolecithal means where you have mostly yolk and only on top portion you have some division going on. So here itself some layers are forming. So they are asking blastocyst is formed where in that embryo. So telolecithal egg, it undergoes meroblastic cleavage, which means incomplete cleavage it will undergo. And because it looks like a disc from the top, it is called meroblastic discoidal cleavage. Okay. So if you remember, two layers are created first, right? So one is epiblast, another one is hypoblast. But where is exactly the blastocyst? Okay, so anyways, I'll show you this one. The answer is primary hypoblast and secondary hypoblast. Okay, primary hypoblast and secondary hypoblast. So what is that? Initially, you have just one layer, which is the epiblast. Okay, we have seen this diagram already, and this is the yolk region. In between the yolk region, you have subgerminal space. Okay, later some hypoblast breaks from the epiblast. So that comes out by delamination. Delamination, we know, breaking into two layers, right? Yes, Sunshine, your answer is right. So this will join together, okay? So they will join together and that will form the primary hypoblast, okay? Later, one more layer will join from the colar sickle. So they will form another layer over here. So it is present between the, so blastocyst, you can see it is present between epiblast and this hypoblast. So here it is present between the epiblast and the hypoblast. Okay. And this is still what is the subgerminal space. Subgerminal space is here only. Okay. Okay, so that's all, everyone. Thank you very much. We will do unit 1 in our next class. Okay.